Hi, my name is Harshita, and my project is on the molecular effect of ionizing and ultraviolet radiation on the Arabidopsis thaliana genome. Over the years, there have been many nuclear accidents, including Chernobyl and Fukushima, and because nuclear power is becoming a significant energy source, there's likely to be more in the future. These accidents release radioactive contaminants that can be extremely harmful to many organisms, including humans. Nearly one-third of the world's reported case of acute radiation sickness, which is radiation poisoning, has been due to Chernobyl. And studies have shown radon emissions to be the second leading cause of lung cancer in the US. However, not all organisms are affected the same way. For example, plant abundance increased after Chernobyl due to the positive effects of land abandonment and human evacuation. Many studies have been done to support and oppose adaptation for plants, understanding the molecular evidence. So ionizing radiation is a primordial stressor, which means it's been around since the beginning of the Earth, so we likely have some level of resistance to it already. However, plants have greater adaptation skills because they're immobile. So either they're going to die or they're going to be able to acquire beneficial mutations and be able to adapt. If we take a look at how ionizing radiation damages DNA, one way is through double-stranded breaks. And these can be repaired by homologous recombination, which contributes to genetic instability and variation. And so this process is promoted by the RAD-A, REC-A, and RAD51 genes, which are radiation responsive. Studies have shown there to be a lower recombination frequency during later years in Chernobyl, which could prove that organisms are able, or plants are able to adapt and become more genome stable, or it's due to the level of radiation decreasing over time. Another way is through radiolysis, which is the dissociation of molecules, and that produces free radicals or reactive oxygen species. These can go ahead and target key proteins and destroy them, increasing oxidative stress. Important oxidative stress responsive genes, including CAT and F3D3, have shown to be upregulated in exposed plants, bringing about an increased level of antioxidants to intervene and prevent these key proteins from being destroyed. This again could be supporting adaptation. If we take a look at some epigenetic mechanisms, in later years, there has shown to be greater mutagen resistance in exposed Arabidopsis thaliana plants. And this decrease in sensitivity has also been shown to have an increase in antioxidants. There has also been an increase in hypermethylation levels in contaminated areas and earlier years because of higher levels of radiation, which could support that plants' primary response to radiation is through hypermethylation, and a return to normal levels can support adaptation, or it could be due to the decreased doses of radiation over time. Specific pathways that have shown to be affected in multiple organisms include the DNA repair, oxidative stress response, and signal transduction pathways. However, if we look at specific organisms like rice, the OSG1RP1 is a radiation responsive gene shown to be upregulated due to radiation that prevents the breakdown of those key proteins. This proves heterogeneity, which means that different organisms are affected in different ways due to irradiation. This study will target the Arabidopsis thaliana genome because this is a model organism, and there have been many studies surrounding whether it is radioresistant or radiosensitive. So the purpose is to compare upregulated genes in this plant for both ecotypes, which are species in different geographic areas that have genetic variation naturally due to other environmental stressors, when exposed to ionizing radiation and ultraviolet, and to determine whether these are potential biomarkers and can contribute to sensitivity or resistance. So initially, I was going to run a gene set enrichment analysis using the ShinyGeo program, where basically I input a set of genes and it compares it to its library of controls to assign them enrichment values and determine their molecular pathways. However, most of my data sets already have genes with enrichment scores, and the molecular pathways aren't as applicable to my purpose. So for most of my data sets, I will be using a differential expression analysis using XLSTAT. Basically, this identifies genes that can be associated with specific phenotypes, so for example, in this case, resistance or sensitivity, due to their expression levels. It also corrects for multiple testing by ensuring that good p-values aren't just due to chance, and it produces these FDR, or false discovery rate values. In doing so, it's able to avoid type 1 or false positive errors. So I had two variables, ecotypes and types of radiation, and each of these have two conditions. So for my 4 conditions, I have a total of 8 databases, and I will specifically be targeting this one that I've already analyzed. So after running the differential expression analysis on this database, I was able to output these 30 genes that have the lowest p-values, so they're all significant, and their expression levels under each of these three conditions. As shown from the graph, ionizing radiation clearly has the most overrepresented or upregulated genes. However, I was more interested for the exposure of gamma ray. So after looking at this chart, I was able to uncover these 4 genes that have been shown to be significantly upregulated under gamma ray exposure. These can serve as potential biomarkers, but I would first need to compare them to other data sets as well. After analyzing the Arabidopsis information resource and understanding the functions of these genes, I've been able to uncover specific proteins or functions that they may be part of. For example, some of them are able to break down chitin in cell walls or serve for polysaccharide building or for a phosphorylation chain and so forth. So for further direction, I would be running my GSEA with my databases without enrichment values and potentially using Biomart for gene ID conversions, and then running those expression values through differential expression analysis to determine my upregulated genes. If possible, I would love to go into a wet bench environment and use CRISPR to knock out these potential biomarkers and determine their association with resistance or sensitivity. This project is highly applicable towards space science because it has long been a challenge to take radio-resistant planes into long manned flights due to high levels of gamma ray and ultraviolet in the cosmic environment. Therefore, we can use gene editing and these potential biomarkers to help tackle this. In addition, orthologs are these genes that are in different organisms but generally have the same function. So we can look for orthologs of these potential biomarkers in humans and determine its level of relativeness towards ARS, or acute radiation sickness. In addition, many agricultural farms near nuclear accidents have had an extreme decrease in crop yield due to contamination from radiation. By using gene editing, we can help to harvest and save the crop yield for over time as well. Thank you, and do you guys have any questions?